Well, nau mai, haere mai. Welcome everyone to Rangahau Rising. I'm Nick Waipara, I'm your MC, but I'm going to open our karakia. You can join that if you want on the app to bless our hui today. Kia hora ti marino, kia whakapapa pounamu te moana. Hei huarahi mā tātou e te rangi nei. Aroha atu, aha mai, tātou e tā, ti hei mauri ora. I have a great pleasure to introduce Dr. Hanareia Ehau-Taumaunu. She's going to talk to us today about her one postdoctoral research. Hanareia hails from Te Tairāwhiti of many iwi hapu on the East Coast. She has been overseeing a PhD at Pennsylvania University and since returning to Aotearoa, New Zealand, as a postdoctoral scientist at Bioprotection Aotearoa. She has been based here in Tamaki Makaurau at Plant and Food Research. She's also been one of the key Myrtle rust, the invasive plant pathogen that's been attacking our taonga, and one of the few or only people I know of who are actually studying the taonga plant Maire Tawake. So I'd like, without further ado, to give the floor to Hanareia and her kōrero today. Welcome, and the floor is yours.
Tēnā rā koe, Nick. Mō tō whakataki I tēnei hui, rā tēnā rā koutou katoa, ko whakahuhui mai nei, ki te whakarongo ki au I tēnei wa. Ko wai tēnei, ko Hanareia Ehau-Taumaunu ahau. He uri tēnei no Ngāti e Pohatu, Ngāti Parau te whanapunu me te Aotearoa. He kairangahau ahau, ki te rangahau marakai, me Bioprotection Aotearoa hoki. Yeah, as Nick said, I hail from the East Coast, but I also whakapapa to the West as well. And I am currently postdoctoral researcher at the Plant and Food Group of the Bioeconomy Science Institute and Bioprotection Aotearoa. And yes, as Nick said, I'm going to be talking everything microbiomes and how myrtle rust is impacting those microbiomes. So to introduce our beautiful taonga that I have been working with is Maire Tawake, Syzygium Maire, but also known as waiwaka and whawhako. It is an endemic swamp forest tree species of the Myrtaceae whānau here in Aotearoa, New Zealand, and it is our sole representative of this genus. But many of its exotic cousins, such as lily pilly or monkey apple, have been introduced to Aotearoa over time. Maire Tawake is a beautiful and distinctive tree that is found throughout the North Island and the top of the South. These raka are typically found in waterlogged soils, on the margins of swamps, or poorly drained areas bordering streams, depressions, and gullies. It has this smooth, white, soft bark with a papery texture on a trunk that lacks a buttress at the bottom. So it's a very straight trunk and it has this beautiful whiteness to it. It has specialized roots, in particular the pneumatophores, which you can see on the slide, that grow out of the ground, allowing the tree to survive in wet conditions. The Maire Tawake blossoms, their flowers, they are white and similar to those of pohutakawa that many of you will be familiar with. They also have fleshy fruits, so they're one of the rare Myrtaceae that have fleshy fruits, and they're very bright red, as you can see in the picture. They are the largest fruits of our endemic myrtles, and they only contain a single seed. And finally, the leaves, which I am doing a lot of my mahi on and have a passion for. These leaves have these kind of like wavy edges to them and are oppositely arranged. The whakapapa and connections of Maire Tawake are significant for many hapu and people across Aotearoa. The raka is used as a rongoa and in dyes, and is ecologically an important mid-story tree in wetland forests. So I have come to love Maire Tawake. A recent study of the population genetics of Maire Tawake by Dr. Colin Bulkwell will show that the raka cluster in northern and southern regions of the North Island, as we can see here on this principal component analysis of about 188,000 SNPs in the Maire Tawake genome. So we have the ones that are clustering north with the northern Taranaki and the Bay of Plenty to those further south on the North Island in the Manawatu and Wellington regions. This indicates that Maire Tawake are somewhat genetically distinct as you moved down the country towards the top of the South Island. Today, the population of Maire Tawake is nowhere near the size that it used to be in the past, mainly as a result of clearing of native forest and drainings of land. This rako is now classified as critically endangered because of its extreme susceptibility to myrtle rust, Austropuccinia psidii, and currently there are limited disease management options for this disease. So we're going to look at the microbiome to see if it could be part of our answer. And why are we looking at the microbiome? Well, really, microbes, those tiny living things that are found all around us, such as fungi, bacteria, viruses, and protists, represent approximately 95%, if not more, of all living biodiversity on our planet. And the community of microorganisms, i.e. the microbiome, live on and in plants. And they are sourced from the environment, such as the rain, the air, and the soil, and can be passed from one plant generation to the next. So the plant microbiome is a dynamic and complex ecosystem that plays a crucial role for disease prevention by directly inhibiting a pathogen through the production of antibiotics, for example, or outcompeting pathogens for resources by depriving it of essential nutrients or space. And they can also, microbes can prime the plant's immune system through induced systemic resistance. Therefore, the plant microbiome really contributes to a plant's overall health and resilience. So I set out to answer a few questions to support the resilience of Maire Tawake in the face of myrtle rust. And so my first set of questions are really about understanding what makes up the microbiome of Maire Tawake leaves and where those microbes may be coming from. To do this, I collected leaf samples from 3 geographically different locations across the North Island, Te Ika Maui, in Tamaki Makaurau, Taranaki, and Wairarapa. Maire Tawake today grow to an average of 16 meters tall. So I had to get quite resourceful when out in the ngahere, as you can see here in this picture of me trying to collect leaves that are way, way taller than I am. I collected asymptomatic leaves from a focal Maire Tawake, as you can see in this diagram, which is the big spot in the middle, and then 9 neighbouring plants, which included trees, ferns, and vines. And those were all within a 5-meter radius of that focal tree, that Maire Tawake that I was sampling. From those leaves, the microbes from the leaf surface were collected and sent back to the lab. Once I was in the lab, I then extracted all of the DNA of those microbes that I had collected, and then I sequenced identifiable regions of bacteria and fungi. The resulting data was processed to form amplicon sequence variants that can be taxonomically identified and analyzed, so we know who's there and what they might be doing. And so what microbes are on Maire Tawake leaves and how did they get there? So here is a bird's eye view of the Maire Tawake leaf microbiome. And we can see that the leaf microbiome is comprised of several fungi and bacteria. Many of these are common leaf inhabitants or common environmental microbes. And in these graphs, the relative abundance, or the proportion to the total, of each fungal and bacterial family are shown here. And you can see that they're actually really similar across the different locations that I sampled from. You can see pink and all, you can see blue and all, and so you can see that there's a lot of commonality in the families of taxa that are present across the country. And also that there's actually a lot of diversity. So there's ones that are have a large relative abundance and those that are small. So we have ones that are very common and ones that are unique. and they're likely all playing a role in this ecosystem on the leaf surface. I then wanted to see if there was kind of like this shared core microbiome that these plants, these trees might have. And so this is when you start to kind of overlap all the different parts of that microbiome to see if there really is something right in that middle of these graphs that they must all contain to hopefully function as a successful microbiome. And so here we're seeing a common set of those ASVs, those regions that I had sequenced across the 3 locations. And even though on the previous slide we just saw that they must be sharing a lot of these microbes, we can see in those middles with fungi with just 21 ASVs and bacteria only with 44, that they do share a very small and limited core, but also contain a mass of others that might not necessarily need to be present or selected by the tree itself. And so there's a lot of kind of multiple factors that are coming into this scenario where the tree can also select its microbes, but it also depends what's else in the environment, and then also the competition between the microbes themselves in the microbiome. And so it's very low overall. One of the interesting points that I want to point out is that between Taumaki Makaurau in the blue and Taranaki in the green, they actually have a similar number of shared ASVs, or even higher than that when you actually put Wairarapa into that scenario as well. And that kind of seems very similar to the trend that we were seeing with the plant genetics, where they were separated into northern and southern populations. So maybe the plant is actually really having a key influence here on the microbiome on their leaves, and that's actually seeing as a distribution across the country as well. The low number of shared ASVs here was quite shocking to see for myself. And I was like, why could this be when I just showed on the previous slide that things were being shared across? And so it could be more that the slide that I showed previously was at a higher ranking level taxa, whereas these ones are probably more at the genus and species level because we're looking for those that are really unique in those sequences. And so there could be a higher diversity of things at the genus and the species level where we're not really seeing them common across, but their families that they belong to actually are shared across these trees as well. And so to look more at those differences between the microbiome samples, I performed a canonical analysis of principle. And this is really to see the differences in between samples. And the results here show that the fungal and bacterial microbiomes cluster based on their location, with Taumaki Makaurau in blue, Taranaki in green, and Wairarapa in orange. These differences are explained partly by the location, but more significantly by the tree's exact location within each ngahere, shown by the high R-squared value here as known as tree site. The beta diversity here is indicating that the microbiome composition of Maire Tawake is shaped by the tree, reaffirming the likelihood of the lower ranking taxa at the genus and species levels being more distinct between trees that we saw on the previous slide. And so that's giving us a really good idea of the leaf microbiome for Maire Tawake by itself, but how does it compare to its neighbours? And so when I looked at the Maire Tawake microbiome compared to its neighbours, which you can see on this slide here, with one bar being Maire Tawake and the other bar being its neighbours, which are those 9 plants that were around the focal Maire Tawake, we see again a diverse number of common fungal and bacterial taxa that are similar in their relative abundance between the two. And that there's actually quite a bit of diversity in terms of their families that are present, and that also increases as you go to the lower levels of taxa. This suggests that there is microbiome sharing occurring between the plant leaves and the ngahere, or they are also acquiring the same microbes from the environment. Here we can see again that the microbiome samples of Maire Tawake are now as circles that are coloured, compared to the neighbours as triangles that are exposed, and they again cluster based on their location. As we can see, all the colours somewhat are close to one another. We can also see that the neighbouring microbiomes in terms of triangles are actually more similar to each other as they kind of come closer in terms of their proximity to one another. The variables of the tree's exact location, so that tree site variable again within each ngahere, doesn't seem to be as influential when looking between Maire Tawake and its neighbours, as we can see here with a lower R-squared value. And so the tree effect really is for the individual trees themselves, but when you increase the number of trees into that scenario, then there likely is sharing, and so there's not as much distinction between them. So the trees within each ngahere are similar to one another, but there are also differences in amongst them, even between Maire Tawake and its neighbours, and between Maire Tawake themselves. When I went to look to see if there was a core common shared kind of microbiome between Maire Tawake and its neighbours, the number of ASVs shared were actually higher than the number of ASVs unique to either host. So this is where we're starting to see that the neighbours are likely playing a key role in providing microbes to the Maire Tawake microbiome. But however, we can see that Maire Tawake still host a number of unique ASVs that we don't see in their neighbours, again, likely indicating that the plant host or Maire Tawake is selecting those microbes in particular, likely to help with its overall growth, health, and resilience. When looking at differential abundance using NCOM-BC to identify microbial taxa, this is really looking to see if there are microbes that are significantly different in their abundance in the different rako between Maire Tawake and its neighbours. I only found a very small number of ASVs that were significantly more or less abundant across both fungal and bacterial microbiomes, as we can see here on these two graphs. Maire Tawake had more abundant ASVs seen in the red dots, whereas neighbouring plants had less differentially abundant ASVs seen in the green. This suggests that some taxa, again, are being selected by Maire Tawake. Out of interest, the more abundant fungal ASVs on Maire Tawake were Ascomycota, some known to be pathogenic. I then used the Sloan neutral community model to understand the processes that are shaping the microbial community. So what are some of the pressures that are on these microbiomes in terms of how they're kind of coming together and who is able to stay and who must go? And this model quantifies the relative importance of deterministic processes, such as environmental filtering, whereas stochastic processes kind of come from more random dispersal. It's more stochastic, it's more random in its nature, likely coming from the environment and not being kind of selected by the tree. And we can see on these graphs that the R-squared value is the very important value that I want you to look at here, and that the fungi with the low R-squared means that it's actually affected more by deterministic processes. So then the plant host is playing a bigger role in the members of the microbiome, whereas the bacteria actually have a high R-squared, which makes it appear to be more stochastic in their assembly. So it has a lot of immigration, migration coming into those populations, and it's just going with the flow rather than being selected for and having a presence of certain microbes through that way. And so now we have a better understanding of the who and the how of the Maire Tawake leaf microbiome. But does that change when the leaves are infected by myrtle rust? For this comparison, I did another set of sampling of the leaves again, however, only in Tamaki Makaurau and Taranaki during the height of myrtle rust infection. Maire Tawake leaves were collected from both asymptomatic and symptomatic leaves based on visual symptoms. And again, the microbiomes of the leaf surface were collected, and then they were sent for whole genome sequencing. So rather than just looking at a single gene to identify the microbial taxa, all parts of their genomes could be sequenced in the hope to gather more information from their DNA. In light green, Sphingomonas in medium green, and Streptomyces in brown at the bottom, with the largest portions, largest relative abundance within the bacterial microbiome. These three genera are again very common leaf inhabitants and or are highly present in our environment. Tamaki Makaurau and Taranaki appear to share relatively the same abundance of their taxa, but there are many low abundant taxa or unidentified taxa that we can see here present in the grey section of the bars. Unfortunately, this is a common occurrence with this type of data, but I do plan to go back and try to reduce this population, this proportion. So really trying to increase more information out of this sequencing data. But here we can just kind of see an overall of what might be happening and actually still seeing common leaf inhabitants, which is great to see. In contrast, the symptomatic leaves, so those that had visible myrtle rust infection, actually lack diversity compared to what we saw on the previous slide. Here, the infected microbiomes, the infected bacterial microbiomes, are greatly dominated by two general bacteria, Pantoa in medium blue and Pseudomonas in light green. Again, two prolific leaf inhabitants and very good at populating and competing with other microbes. And so here is the view of the last two slides in one. So you can really visualize the stark contrast between the bacterial microbiomes of asymptomatic leaves versus the symptomatic. We can see that both the richness and evenness decreases, and it's showing that the fungal invader, myrtle rust in this case, can really influence the entire microbiome, not just the fungal members. And when I look at these whole microbiome samples for the fungi, fungal reads were very small in proportion to the overall reads that were sequenced. Fungal genomes are typically larger, and the bioinformatic pipelines are actually better equipped for bacteria rather than fungi. Yet we can still take a look at to what is there, kind of to identify maybe some trends of who is there and what might be happening when myrtle rust is present. The coronapots that you're seeing on the slide here represent an individual sample and do not show relative abundance. Rather, they're the percentage of reads identified, because I'm still trying to find the best way to get more information out of these reads. But I'm hoping to give you an early glimpse into the diversity of the two main fungal groups between asymptomatic and symptomatic leaves. The slide shows a number of genera in the Ascomycota phyla that are present in these two samples. They host similar fungal classes, however, the lower ranking taxa do differ. So it seems like the Ascomycota diversity increases in the presence of myrtle rust, because we can see less kind of boxes and names on the asymptomatic, and you can see that kind of increases when we get the symptomatic leaf data. For Basidiomycota, which is another phyla of the fungal kingdom, which includes rust pathogens, such as myrtle rust, we see this shift in the percentage of reads that have been classified as Puccinia at the genus level. And just for your information, myrtle rust used to be known as Puccinia psidii before it became Austropuccinia psidii, and the databases have likely not been updated, so we can kind of assume that Puccinia here could be representative of myrtle rust as well. And so we see that there's this, in the asymptomatic, there is a small portion, not to say it's myrtle rust, there could be other Puccinia out there, but then we get this dramatic shift when we go to the symptomatic leaves, where there's a larger portion, a larger number of those reads identified as Puccinia. And so it's really showing, again, that impact on having myrtle rust. Yes, it might be there on an asymptomatic leaf because you can't see them when they're very small spores. But when we've got lots of pustules, obviously they're going to be a larger portion of that microbiome. We can still see the trend where the overall diversity of the Basidiomycota increases between asymptomatic and symptomatic. And this could be an individual effect, but looking at the other samples that I've sequenced, this seems to be quite a relatively good trend, that diversity is still increasing between when leaves likely do not have myrtle rust to those that definitely have myrtle rust present. So how do these fascinating microbiome insights help with the resiliency to myrtle rust? First, knowing who is there enables us to identify potential antagonists of myrtle rust that could be researched for their biological control abilities. When I dove deeper into the fungal genera from both datasets, I found that many are known to be antagonists of myrtle rust at the center of origin in Brazil. So that is where myrtle rust has come from. So we may already have part of the answer in our own backyard. Secondly, targeting the pathogen is just one part of the equation. We also need a healthy ecosystem and healthy trees to enable Maire Tawake to flourish. So understanding what that might look like can help support our conservation efforts by optimizing the environmental conditions, and in this case, the microbial conditions, to prevent or reduce the impacts of myrtle rust. And lastly, we can push this further by knowing not only what Maire Tawake needs, but how the whole ngahere can support its protection from the myrtle rust. And so my conclusions so far, as I still have plenty of data to go through, but for the Maire Tawake microbiome, the overall microbial richness and evenness within samples are quite comparable, but the composition of the low-ranking taxa between those samples appear to be distinct. We also could see that there's a lot of sharing between Maire Tawake and other trees in the ngahere. However, the exact location and plant host itself play a key role in the unique taxa found on Maire Tawake leaves. We also saw that the formation of the fungal microbiome is facilitated by more deterministic processes, such as the environment and biotic factors, compared to bacteria, which just seem to appear to have a more random construction within its microbiome. For examining what happens to the Maire Tawake microbiome when myrtle rust is present, preliminary results show asymptomatic leaf microbiomes are comprised of typical leaf inhabitants and can be quite diverse for both bacterial and fungal genera. Symptomatic leaves are heavily impacted by myrtle rust, with the Puccinia genera having a larger number of those sequence reads. And overall, there were actually more bacterial and fungal taxa identified in this data set. And just seeing that diverse kind of change between asymptomatic and symptomatic leaves, likely indicating opportunistic microbes kind of taking advantage of the circumstances. And so with that, this mahi could not have been done just by myself. There are many people and rōpū that have been involved. And so I just want to give a big mihi to Ngāti Tamaoho, Ngā Rohine, and Ngāti Kaununuku Wairarapa that allowed me to undertake this research on their taonga rākau in their rohe. Also to Waitmore Farms, Christian Farms, and the Department of Conservation who enabled me to get access to these rākau. They're not the most easy to get to, let alone to get enough of them in the same place. So these people were really generous with their time, enabling me to access these rākau. And then lastly, to Dr. Nick Waipara, who has been a supportive supervisor throughout my postdoc journey, and also to the Plant Pathogen Environments group, which is who I work with on a day-to-day basis, who I've pestered about how to do field work, thinking about the microbiome, and any other considerations that I've really just needed someone to springboard ideas off. So thank you all so much. And then lastly, to Bioprotection Aotearoa and Plant and Food Research Group for enabling me to come home and to focus on some mahi that is very dear to my heart and my people as well. So tēnā rā koutou katoa.
Kia ora. That was awesome. Awesome mahi. The pātai we're going to have in the chat. So if you want to ask a question of Hanareia and her fabulous research, please put in the chat. Some comments first from me. So kia ora for that hard mahi, and really interesting mahi. The that grey area, for example, in the asymptomatic bacterial microbiome, does that grey area that you're having to, a grey area, very apt, having to do more deep dive into the sort of molecular identification of the groups there? Or do you think there might actually be unknown, or so you might have to go up the classes? I mean, is it is it is it that age-old problem that there's not much knowledge around the microbiome, especially of taonga in Aotearoa, that it's data deficient, or is it perhaps just that needs a different technique to identify them?
Personally, I think it's all of the above. Databases are only as good as what's in them. So if we don't know something exists, then that's not going to be in the database. So there are those unknowns. Also, a lot of taxonomy changes all the time. So updating those databases to be accurately of the systems that we currently use as scientists can also play a role. And then also there is the technology that we have. As I said, the fungal are really hard to pick up when you're doing a whole metagenomic sequencing of the microbiome. So you're not just sequencing isolates that you've been able to culture and things like that, which are a bit easier because then you know you have a single source that things are coming from. This type of sequencing is just taking everything from that sample. And the host plays a key role because that can always taint or be a large portion of your sequences. In this case, mine is probably a bit lower because I've just taken the microbes off the surface. But yeah, it really is just, there's a lot of things that we can improve on, both technology wise, database wise, and then also just biology wise as well.
Kia ora. Oh, well, good. Best wishes, Namahi, for that, for that further work. I've got a kia ora, Nicola Day. He's got a great question here. Great talk, Hanareia, of course. I'm interested in the relationships with the neighbouring plants. Are the patterns you see similar to those of other plant leaf microbiomes?
Yes. So there's quite a bit of work done, mostly in crops, of course, about this neighborhood effect, which is, are your neighbors sharing their microbiome with you and vice versa? And there is quite a bit of sharing, and there's a lot of different factors that go into that. So it can be affected by plant age, the size of the plant, how many leaves it has. Again, this is also probably happening at the root level as well, so below the soil, in place like that. And they can see that there's a lot of kind of sharing in that. And then the environment itself also plays a key role. So you might get a new microbe kind of landing on your leaf, but if it rains, then it might not stay. And so there's all these different factors that go on with the leaves because they're just exposed to everything and anything. But there are definitely a lot of other researchers out there that are looking into this effect. That's also the same in forests, mostly when it's like a monoculture that's next to something else. You can also see similar things there. And then also, I found a paper recently that looked at neighbouring forests next to a vineyard, and they could see that the forest next door was actually sharing microbiomes with the vineyard as well. So there are definitely a lot of sharing. I don't know if the patterns are exactly, and I feel like that's going to depend on the location itself. Because yes, we can assume a lot of microbes are everywhere, but there's a lot of unique microbes to say Aotearoa compared to say somewhere else in the Northern Hemisphere. So yeah, plenty of trends to look into. And it was interesting to see that this is playing a key role in the microbiome sharing within our own ngahere.
Kia ora. Kia ora, Jay. Well, it wouldn't be a webinar without Jay. Are you noticing any regional differences in susceptibility to myrtle rust, indicating genetic sources of resistance relationships with the microbiota?
Unfortunately, along my treks across the motu, I found myrtle rust everywhere. And particularly with Maire Tawake, it is one of our most susceptible trees to myrtle rust. So it's pretty much up there with Ramarama, which many of you might know. So I don't know if it really is playing a difference in susceptibility to myrtle rust. I do have another data set that might be able to help answer this question in a way. So I have gone out and sequenced the leaf microbiomes of the other Myrtaceae, Arako in this family, because we know that they definitely do have a spectrum of their tolerance to myrtle rust. And I want to see if the microbiome is playing a role in that. So are they different? And if they are, what, how, why? And then I'm going to take it a step further from sequencing those, and that I've actually maintained some of those microbiomes, and I have applied them to Maire Tawake seedlings to see if they have a protective or what's it called, or like a treatment effect on those seedlings. And based on my stats, which I'm still working through, it looks like Fijoa and our Metrosideros perforata have better effects at protecting against myrtle rust. So there likely is something about the microbiome happening, but you'll have to wait and see to find out more.
Oh, that was part of the problem when you first started your mahi, was the appearance of, well, the rarity of Maire Tawake actually in the environment, even with the whole gaggle of botanists who know every tree in the rohe, find it struggling to find the rako, but in a serious sort of number. And then second, by the time you got there, the rust was already spreading and was quite debilitating, wasn't it? So that just shows you how vulnerable A, the taonga is, and B, what you've been having to grapple with. It's been going as you've been going. So well done for managing that part of your mahi. From Stuart, kia ora, Stuart. Were the neighbouring trees sampled also rust host, other rusts, not rust? So were there other rusts found? If so, were they carrying any of the potential known rust antagonists, e.g. Sphaerolopsis?
Yes, that's very interesting. Because unfortunately, because of the region that I sequenced for the fungi, which was ITS1, it is a region that doesn't really well identify rusts in general. So I can't really tell you if the neighboring trees were rust hosts based off that. I think that's definitely something that I'd love to follow up with and maybe do a more of a metagenomic kind of sampling and just seeing everything that's there, or even just using ITS2, which is a better region for identifying them. But I did find that they, again, were also carrying those other known antagonists that I had listed on the slide. So those weren't just on Maire Tawake themselves, they were also on the neighboring trees as well, or plants. So yeah, can't tell you whether they were rust hosts, even, you know, some of our native rust or anything like that, but they definitely have those antagonists there as well. So they could also be another source or a way to, you know, kind of amplify those particular microbes to support Aurako.
Actually, on that note, you know how we have the Kodi rust and the Putskawa rust that were already known. Has there been any sort of, I don't suggest microbiome investigations, but any of those sort of early botanical looks at potential native leaf rusts or anything similar on Maire Tawake in the past? Probably
To
not,
my
but I
knowledge.
thought I'd ask.
No, to my knowledge, not really. Again, we haven't really thoroughly studied most of our Myrtaceae species here, and at least in a microbiome sense, I don't think so either. So yes, but I think I can probably easily go into the data sets and actually to see if I can target those gene regions from those only if they've had a genome kind of done as well. So yeah, I think we definitely need more research and more awhina for our Myrtaceae species.
Yeah, got it. The other part is what's the closest extant tree relative to Maire Tawake?
Oh, I should know this, Jay. Um,
Well, it's a Syzygium, so I
yes.
don't know anything other than that.
So so it's the only one here, and I think its closest relatives are actually in Australia. There are way more Syzygium um, plants in Australia. Like there's so many, and they're also very different from ours as well. They have ones that can be in the swamps, through to the cliffs, and everywhere in between. And so I'm not entirely sure of the exact species that is its closest relative, but it definitely is those that are in Australia. So it was likely when we were once many thousands and tens of thousands of years ago when we were together, they would have likely split, and then ours has become a very specialized rako in the swamp.
Any more partai? I've got the other part of the is the mātauranga. What has been the I guess the knowledge that you've learned locally from the different iwi hapu whānau that you've been visiting while you've done your mahi, particularly I remember you visited the marae at Wairarapa, but have you found the interest in the mātauranga around Maire?
Yes, I think many of us are still in the process of reclaiming that mātauranga with Maire Tawake. It was a very great wood for building buildings and things like that. So it kind of, in its history, has been moved from a rako that's ecologically important to something that became a resource, and then something that's somewhat become forgotten from the general public. And so a lot of the mātauranga is looking at around the kupu in the ingoa and kind of connecting that to whakapapa. And then also just noticing that usually, Maire Tawake is typically found with other rako. And then they have their whakapapa from tāne. So the likes, I always used to see, pukatea, kahikatea, and a few others. Whiki, the fern is always found, particularly for the northern populations with it. So they have a lot of whakapapa connections in terms of the rako that are in the ngahere with them. And so that's another point of link. And then there also is the kind of mahi around rongoā. So they would use the bark as a rongoā. The berries are also nutritious. They have probably one of the highest levels of antioxidants in our country, which a lot of people don't know. But those small little berries are such a cool kind of source of nutrients that our tupuna probably would have feasted on, because thinking if you have a mast year and you had all of these red berries on a Maire Tawake, you would be fighting the birds, of course, for those. So there's a lot of kind of mātauranga in that as well, too. So yeah, there's a lot of mātauranga talking to different people. I think they're trying to still make those connections and think about how we can create that mātauranga. And my hopes in this is kind of adding to that mātauranga from the microbial world. So yeah, hopefully it'll continue and won't end with me and a few others that are currently doing this type of research.
Yeah, because I remember the kōrero with Waikato Tainui, and their loss of Maire Tawake from their rohe, and their mahi is around restoration and includes restoration of actually the cultural and the knowledge, the mātauranga and tikanga that wairua that surrounded those taonga that have gone. So it's a not only a biological restoration but a whole cultural one too, and the tragedy of that loss that may never come back if those who knew that have gone. So that's one of those parts of your mahi that's really important that you've been doing with the local iwi. We've got another question here in the chat. Myrtle rust is a particular threat to younger tissues, young trees and seedlings, right? So if you notice any signs of effects on the next generation of Maire Tawake in your sampling trips, so I guess you're seeing that expression, that disease expression.
Yes, I definitely see the disease expression. And I would come across seedlings, that have obviously just formed once the fruits had fallen to the ground. I can't say that I saw many that didn't have myrtle rust on them or didn't have um, things that appeared to be kind of like old symptoms. Over my time, I start to learn like, no, this isn't Maire Tawake. It's probably just some other microbe that's living and taking an advantage, or it's just another pathogen that we don't know about on Maire Tawake. But many of those seedlings definitely were infected or had kind of visual symptoms that looked like it had been infected from the last season. But then you can also see, I could also see for the very larger, older trees, you can start to see a lot of them that I visited were already kind of having dieback on those Darko. Some, I could just see that it's likely not going to survive. That doesn't mean it's just myrtle rust. It could also be environmental conditions and things like that as well. But then as you saw probably in my first slide where you saw the myrtle rust kind of prolific on that leaf sample that I'd taken the picture of, that also includes myrtle rust on the actual flower buds that are coming through. And flower buds once they open, they will seed, and then it takes about the fruit a whole entire year to actually become ripe. So if myrtle rust is already attacking those flower buds before we can actually get a flower to be produced, then you're not going to have the next generation coming through. You won't have those seeds that can populate in a new place, after the bird's taken them somewhere else. So unfortunately, I was seeing those signs very vividly across the kind of spectrum from seedling to very mature trees dying. And that's just not my reflection. I think that's the reflection of many researchers who are trying to protect Maire Tawake and mutae them as well.
And lucky last, kia ora, Marion. The shift in the microbiome due to myrtle rust, is that also explained by the leaf plant response to the infection? Because a common plant response is a leaf abscission. So
yes, it could be explained by the plant response. Although I've just seen leaves riddled with myrtle rust as well. So I think it's because the rust forms those pustules and I wasn't being discriminative of what was on the leaf surface. That might also sway my sequencing as well, more towards being representative of those rust pustules versus everything else that was on the leaf surface. But yes, the plant obviously can try and respond, but yeah, usually it's just that kind of formation of the leaf trying to restrict the movement of the rust. But if you get enough of it there, I don't think that leaf has a good chance. And I was only sampling the leaves, so if there were no leaves there for me to sample, I wasn't sampling that tree. So there could be other parts of the tree as well that might aid, because we know that the myrtle rust infects the stems as well. It's not just the young leaves. So yeah, there's a lot that I haven't fully understood, and I think that we definitely need to do more to really understand this as well.
ngā mihi. Well, ngā mihi kia koe, kia ora. Thank you for this amazing kōrero. Ho mai te paki, a virtual clap from everyone for the kōrero and answering those amazing questions. So thank you, everyone. Thank you, whānau, for your pātai today. It's actually really exciting knowledge amongst the devastation. And I think kia ora to you and your hard mahi over the last couple of years, really running while the rust is running too. So ngā mihi to you and your hard mahi and dedication to get through quite a difficult, I guess, biological invasion while it's happening. You're doing your mahi, and it's really important for our taonga that taonga like Maire Tawake are explored by what you're doing. So kia ora, and I'd like to thank everyone today for supporting Hanareia and her mahi, and just by being here and listening to her whakaaro. So kia ora to you all. And I also want to highlight that if you know people that haven't been able to turn up today, or you want to go back and look at those amazing the results coming that are being extracted out of those leaf extracts. Today's kōrero is video, so you'll be able to go back and look at that, as are all the Rangahau Rising webinars. And also looking forward to next month's Rangahau Rising with Dr. Alexa Byers to see what she's been up to out in the motu as well. So kia ora again, Hanareia. It's been brilliant to have you here today, and kia ora to everyone here who've supported us in this kaupapa. Look in the chat for lots of links that the wonderful Zoha and co have been putting into. You can link into the latest newsletter, all about Hanareia, and there's lots of other things that you can link into. So kia ora, and we look forward to you joining us next month. Ka kite anō, te hei mauriora.